to the pesticide. When Darwin looked at evolution, he noticed it as kind of like a tree, and so that we all have a common ancestor, and that these populations diverged to give us all the organisms that we have out there. And then we'll go over those. We'll do what's called the march through the phyla, which I think is really fun to look at these kind of different diversities. But again, Darwin stuff was sometimes kind of hard to understand, and sometimes there was a little bit of kind of ambiguity into why pick one rather kind of than the other, right? I mean, there's a number of arguments. In modern biology, we see the fusion of genetics with kind of evolutionary biology that we have there. And so this led to a little bit of a change, and I'll try and talk about that a little bit now. And so population is a group of individuals of the same species living in the same place at the same time, same as Darwin. It's the smallest biological unit that can evolve. And so again, we have two dense populations of trees here separated by a lake. How are they going to mate with one another? Here in the United States, I'm from Chicago, the East Coast. Here's us here. Here's LA down here. Again, organisms tend to kind of congregate together, and so here we can see where that congregation is. The total collection of alleles in a population at any one time is what we call the gene pool. When the relative frequencies of alleles change over a number of generations, evolution is occurring on a smaller scale. This is kind of what we call microevolution. And so again, evolution is this change in the gene pool kind of over time. What we see is evolution abounds in populations. So not all variations in the population are heritable. Only a genetic component of variation is relevant for kind of natural selection. And so variable traits in the population may be polygenic, resulting from the combined effect of several genes, or determined by a very single gene. And again, when we were looking at genetics, we looked at this polygenic effect in things like skin color or height, etc. Polygenic traits tend to produce phenotypes that vary more or less continuously. Single gene traits tend to produce only a few distinct phenotypes. And so here is basically color of snake of various garter snakes, and we can see this here. And then we have our own variety of garter snake in the San Francisco Bay region. Sources of genetic mutations result from basically mutations on all of these. It's only going to come random mutations or induced mutations, and then sexual recombination, which is the reshuffling of alleles during meiosis. So that's one of the reasons why we went through what meiosis is. For any one gene, mutations alone has little effect on a large population in a generation. Organisms with a very short generation pond, however, can have this genetics work on it relatively fast. A gene pool is a reservoir from which the next generation draws its genes. Alleles in a gene pool occur in certain frequencies. And that is, if we have two different alleles, P would be for the frequency of one, which would be the dominant one, and Q would be an example of a recessive allele in a population. And there's a calculation that would give us this. And so again, here we have in this gene pool, we have dominant red, we have recessive white, and then we have the heterozygote, which are the kind of the pink flowers. And again, when we look at this, I'll just kind of go through here. Is we have a population on here, and again, what this allows us to be able to do is I'm going to go through there. What allows it to be able to do is determine the allele frequency of that, and I'll give you that calculation in a little bit. So sorry about that. I'll go through this real quick. Previous. And so it's a Hardy-Weinberg formula that allows us to determine these frequencies. And again, Hardy-Weinberg allows us to determine the relative frequency of some genes like PKU, phenylketonuria, that affects 1 in 1,000 people. And again, we have our dicola, which I like a lot, contains phenylalanine. And then we'll be able to see that in a gene pool. The mechanisms of evolution include what we call genetic drift, gene flow, and natural selection. So in the Hardy-Weinberg equation, basically we're going to have a population that's stable. Once a mutation occurs in the population, it will remain stable. However, we have this thing called evolution, where we have basically one phenotype or another is accepted in a population. And so that's what we're going to talk about here. Genetic drift is a change in the gene pool of a small population. And this is basically due to chance. And so if we have those flowers again, and we see those flowers go, they're going to be consistent. But if we have something that's eating, or a disease that's killing all the white flowers, eventually we're going to have only the red flowers, and the white flower allele will disappear. In a bottleneck effect, we have an example, this is an example of genetic drift as well. We have a drastic reduction in population size. So in a bottleneck effect, what happens is that through chance, we have a smaller population coming from a larger population. Not all the alleles in the gene pool are going to survive that. I'm calling it a selection, but it's basically just a chance choosing that's occurring. And again, this occurs in populations like the cheetah, etc. We have the founders effect, which is when a few individuals that colonize have a small effect, a strong effect on that. We've seen this occur in a number of human populations as well. Gene flow is the genetic exchange with another population. This tends to reduce genetic differences between a population. And so, again, what we're looking at is this what all of us may kind of look like, is that basically as we see continual mixing of genes of the different races, that we will get an individual who is kind of common to all of those. Natural selection is cause of microevolution. Only natural selection promotes adaptation. And so again, we'll have changes in allele just by mutations. And so in order to look at that, we need to look at a concept that we call fitness, which is the contribution an individual makes to its gene pool. And so again, this individual here has a longer beak. He's able to go down deeper into the flower, and he was able to get more nectar. And when we look at natural selection, we have a number of different types of natural selection. So in directional natural selections, we have a whole population moving towards one. In this case here, we're having it moving towards a darker mouse. In disruptive selection, we may have selection of both the lighter mouse and the darker mouse, in which case we get rid of all the minerals. In stabilizing selection, we're getting rid of both kind of the extremes, and we see kind of things like that happens. Sexual dimorphism is a distinct appearance between males and females. It's not necessarily associated with reproduction or survival, and again, it's who you kind of prefer to mate with in sexual selection. So again, here we have the sexual dimorphism in finches, where the males are much more colorful than the females, and here we have big, strong males with big horns, and basically the horns are just used to kind of work with one another. Does evolution occur in human beings? Yes, it does. We have a number of different examples of that, and sickle cell anemia is an example of that. This is a genetic disorder. It affects roughly about 1 out of 400 African Americans, are basically people of Mediterranean descent. And what's happening in here is the cells are producing an altered protein that allows them to be sickle. And again, this used to occur in areas of the world where basically we had lots of areas with a high malaria prevalence. Okay. So what we want to see in basic, we have observations that certain organisms overproduce offspring, and these offspring have individual variations. And what we see is by natural selection, we see an unequal reproductive success in these individuals. This is the Hardy-Weinberg equation. This is the frequency of homologies of one allele, frequency of homology of the other allele, and this is the frequency of your diploid. Sorry, your heterozygotes. Again, this is always going to be 1. And so these frequencies will remain stable unless something else acts upon them. That something else would be evolution. And here we're looking at evolution, the directional selection, disruptional selection, and stabilizing selection.